Hello, Dr. Ribas. We're doing our presentation on gene therapy with Austin, me, Emily, Min, and Lucas.
All right, so what is gene therapy? So basically, gene therapy is just the editing and cutting of a desirable gene to treat a disease or cancer, or hemophilia. Right? Common types include gene addition, like AAV therapy, shRNA for silencing. Then we have CRISPR and TALEN for editing, and CAR, TCR, and TIL for adoptive cell therapies. And then we also have stem cell transfer. All right. So first gene therapy we have is CRISPR-Cas9 therapy. So this acts as scissors to basically cut a gene and knock it completely down to reduce protein encoding inside of a cell that will also reduce the tumor growth in the microenvironment. And as you can see in this little diagram, it's guided by guided RNA to cut in a specific portion of the gene. So types of Cas9 include SA-Cas9, SP-Cas9, and MCas9. They're all different by their size and kilobases and their position activation sequence in the gene. All right. Okay, so practicality of Cas9. So what is it being used and what is the efficiency in current cancers? So right now, researchers are actually looking at how it is more effective and efficient to treat brain cancer than to just use regular gliomas. And right now, it is actually the most efficient gene knockdown therapy, more efficient than shRNA, and it is also delivered by AAV. And it's better to use because of its high specificity in the gene.
And then we have shRNA gene therapy, which is a way to turn off specific genes by delivering DNA that makes short hairpin RNAs, which is shRNAs for short, into a patient's cells. And this will result in a long-term stable silencing of the target mRNA rather than temporary fix. And then this diagram shows the steps. It starts off with a shRNA plasmid or a lentiviral vector that delivers a new piece of DNA into the cell. And that DNA is transcribed into shRNA, which folds into a hairpin shape and has a sense and antisense strand. And the cell has an enzyme dicer that cuts the shRNA into siRNA, which then guides the protein complex RISC and is like a map for the RISC to recognize and bind to the harmful mRNA. And when it recognizes the harmful mRNA, it will cleave and destroy the mRNA that is causing the disease, making the disease causing proteins. And so it turns off the harmful gene effects even though the gene is still there. And then some diseases that shRNA gene therapy can help treat is HIV, Huntington's disease, and liver cancer.
So TCR therapy involves a T cell which is being modified by giving it a modified TCR or T cell receptor gene, which allows it to recognize and destroy cancer cells. And this mainly targets cancer cells from solid tumors such as synovial sarcoma or uveal melanoma. And this is due to the modified T cells recognizing the internal tumor specific proteins. The steps involve T cells being obtained from the patient's blood, the modified T cell receptor gene being added to these cells via a viral vector, and these modified T cells multiplying and eventually being added back into the patient. So, how does the TCR gene actually give the T cell the ability to kill the cancer cells? Basically, the TCR gene allows the cell to recognize certain cancerous proteins on the outside of the cell due to the specific HLAs, which is a human leukocyte antigen that presents the internal cancer cells onto the outside of the cell. And the T cell will then bind to this cancerous protein, allowing it to kill the cancer cell. An advantage to this therapy is that many cells, billions of cells, are able to be made outside of the body and then added back in. And this type of therapy targets cancers with intracellular proteins, which has a greater range than CAR T cell therapy, which targets surface proteins. But a major con is that there are only a few different modified T cell receptors available, meaning that not every patient can receive this type of therapy because the T cell receptor must be compatible with both the HLA and the type of protein that is on the surface of the cancer cell. And since there are thousands of different HLAs and some of the proteins that are inside of the cell actually cannot appear on the HLAs in some cases, not every patient is compatible with this type of therapy.
Next gene therapy would be hematopoietic stem cell gene therapy, where we're editing stem cells to rise to healthy ones instead. And so I'll go through the steps. So the first step of this would be the patient's own stem cells are harvested either through bone marrow extraction, or leukapheresis. The next step would be where the stem cells are modified outside of the patient, and then they modify them to like produce healthy cells, basically. Next step, a working copy of the gene is inserted into the cells. So whatever is faulty with the stem cell, like what gave rise to like bad cells, then this is where the researcher uses an ex vivo approach where they edit the cells and then they cryopreserve them for later use. Next step would be removing the bad stem cells from where they extracted them or where they are in the body so that we can replace them with good cells. And then the last step would be actually re-infusing the healthy stem cells, and basically they will self-renew, and then the stem cells will produce healthy blood stem cells. And then these are actually able to cross the blood-brain barrier and potentially achieve a therapeutic effect in the brain. And this is specifically for blood cells, but it could be used for other type of cells too.
AAV treatment, this is an adeno-associated virus. What it does is it binds to the cell receptor, does endocytosis into the endosomal escape with a nuclear entry. Basically, it's transmitting the DNA in the virus to the DNA in the cell. And then usually because it remains episomal, expression can be long lasting. So things like our eyes, our muscles, our neurons, things that really don't replicate too much is what is most effective here. And you can have varying degrees of invasiveness, such as local, so just placing it on the eye for the retina, intrathecal for the CNS with the spinal cord, and then you can have intravenous for liver and muscle doses that are more intense and extreme. And then this is one of the more understood aspects of gene therapy. There is very good safety with this, as it is non-pathogenic, low inflammation with lower doses. Expression is very long, and then with the genetic outcome, so 2+ years without needed treatments, new treatments are coming back for more. Wide range of capsids, so the capsules containing the virus and the DNA. So it can be specific for different diseases such as vessels, brain matter, nerves, muscles, tissues, all that. This is very approved. There has been many test trials on it. Even though it is a very expensive procedure, there is people who do try it and they're getting more information as it goes. So hemophilia A and B, cardiovascular issues, inherited retinal disorders, and Parkinson's disease are some of the diseases that come up. And some results of gene therapy are, so hemophilia A and B are one of the ones that use AAV gene therapy treatment, and this is intravenously admitted into the liver for both hemophilia A and B, but the difference is they use a technique called SPARC that allows more 5 to 8 folds for the coagulation in chromosome 9 compared to chromosome 8 in hemophilia A. And the results, it's promising conditions, but there's also risks with any procedure. They may develop some fever, low blood pressure, and strong immune reactions because this is a virus after all, and our bodies do reject if it is feeling threatened or trying to take over. With long-lasting antibodies to the vector, 30 to 60% of patients develop T cell responses that can raise the liver enzymes, and then with higher doses, we do have a higher risk. And if it is not treated promptly with steroids, basically, these can reduce the therapy's effectiveness. But participants that did receive the treatment with recovery time, they did express fear that they were worried that the treatment would stop working after a while and they needed support groups, but they did not express concern with long-term consequences of their overall health. Other than that, they have been feeling like they have a sense of freedom and their life back again with gene therapy. And some benefits are single infusion, expression factors in 8 and 9 chromosomes, at least 4 years without having to go back in, and 8 years for hemophilia B, reduction in bleeding episodes, no need for prophylaxis, and improvement of health. But the limitations are the infusion effects, the high variability with how the cells respond, immune response, especially now that we're putting in an outside source into the human body, AAV antibodies, limitations are the infusion effects, the high variability with how the cells respond, immune response, especially now that we're putting in an outside source into the human body. AAV antibodies in children are excluded. This is a very costly treatment and not everybody can afford it, but there is long-term durability and treatment and side effects due to integration risk with this procedure.
Okay, so conclusions. There are a variety of gene therapies, and there's so many more coming around in the world, and they're on the rise right now. And there's a lot of people that are researching them to see what their effects are on different genetic diseases. Obviously, the most common, hemophilia, and cystic fibrosis, achondroplasia, stuff like that. The biggest takeaway is that even though that there are a bunch of different ways to treat these cancers and these different diseases, right now, gene therapy is becoming one of the most studied and researched therapies that are trying to get into clinical trials and will most likely be a great therapy in the future. And this is our works cited. Thank you.